Well, there is. Good morning, ladies and gentlemen. Today, your presenter, Nadia Emad, is going to talk about a very interesting topic, which is where the human were able to solve the puzzle of our DNA for birth. In this presentation, I will try my best to cover all these outlines that are presented in front of you. So, shall we start our journey? Genome editing. Genome editing is a modification of any specific sequence of our DNA that causes health diseases. Scientists use it to insert, modify, or delete the sequence. All kind of living things need genome editing. In agriculture, we use it to enhance the crops to be more edible to tolerance as in wet. Also, humans use modification of gene editing in treating and preventing diseases like diabetes. Also, we use them in animals to try to investigate more about the diseases and how infection affect the human. Over the past centuries, genome editing and the scientists weren't discovered genome editing yet. The first, they discovered a traditional ways and random ways like crossbreeding, protoplast fusion, and mutagenesis. Then come the innovation of genetic engineering. After it, the scientists were able to discover the gene editing, which are able to specify any sequence in our DNA. The first gene editing technique was RABV. Then came the meganucleases. After it, the zinc finger, and then the talon, and the most and recently used CRISPR. The technique of gene editing depends on the binding that happens between the DNA and one of the three nucleases of gene editing. This binding results in induction in the double-strand break in the DNA. After modifying our gene, we leave the DNA to naturally repair itself by either non-homogeneous end joining or homogeneous pairing joining. Now to the most interesting part in our presentation, CRISPR. CRISPR is short for crystal regular interspace short palindromic disease. Repeat. CRISPR consists of Cas9 protein that can cut the DNA and the guide RNA that's able to recognize the sequence. First, the scientists identify which sequence use or cause the health disease. Then they create the RNA as in the video. The complex of the RNA and the Cas9 protein are then identified to the target cells where the RNA starts its journey. When the RNA identified, they cut. The scientists are now able to modify, cut, or insert another sequence. CRISPR has many uses. The first is editing of crops to be more edible, producing a powerful antibiotics, as we all know we are short in them. Third is alerting an entire species instead of alerting one or two genes, and then treating of diseases. As in 2017, the scientists were able to target the commander center of the cancer that caused abnormal growth of the tumor. And lastly, creating a designer embryo, as in 2018, scientists in China were the first to create the first two human twin girls by using CRISPR. So now that we all know about gene editing and CRISPR, do you think that gene editing is spelled the end of all diseases like cancer, diabetes, and HIV? Well, I will leave you to your imagination.